Hi guys, I just wanted to make a brief video to debrief for the pre-lab for the first in-person lab, which is going to be the cell experiments. So the first part of the pre-lab was a cell counting with a hemocytometer. So the tradition is to count cells in the 5 boxes that are the corners in the middle box. The other tradition is to decide if a cell is in the box is to only count cells that are clearly in the box, but also not touching the bottom or right edge. So in that case, I counted actually 32 cells, and you may have only counted 31 if you decided that this questionable guy was not in the box. So 31 or 32 cells is acceptable. So we want a cell density calculation because this is handy when we're working with cells. So you had to calculate the number of cells in a volume. So the dimensions of the hemocytometer were given to you. Each one of those boxes is 1 millimeter squared with a height of 0.1 millimeters. So all 5 of the boxes are going to be 0.5 millimeter cubed, right? So you counted 32 cells, and in the volume of all 5 boxes, that gives us 64 cells per millimeter cubed. And if we convert to more meaningful units, just making sure that your units work out, you would calculate 64,000 cells per mil, or 62,000 if you decided to only count the 31 cells. So the second question was, given your calculated cell density, how many milliliters should you use to resuspend your cell pellet so that you have 10,000 cells per mil, right? So we calculated 64,000 cells per mil. So we actually want a more dilute cell suspension. So what volume should you get? Going back to Gen Chem, with C1V1, C2V2, you can do this calculation. So our C1 is the calculated concentration that we started with, 64,000 cells. Our desired C2 is 10,000 cells per mil. And then V2 is whatever volume of cell media that we want to end up with, right? So how do you find V1? So to find V1, that requires careful reading of the procedure. So depending on when you looked at the procedure, it's either 2 mils or 2.5 mils of cell solution that you're going to be putting into your 2 different tubes, right? So you would use this number to calculate, and you would end up with 12.8 mils of solution. But really, we only need to end up with about 4 mils total, or actually 2 mils for each one of these. So we'd be wasting over 10 mils of media. So a better question would be, what portion of your cell solution do you need to pellet in order to end up with 20,000 cells per pellet? So remember, 20,000 cells per pellet, where does that number come from? So that comes from, we want 5,000 cells and we want to put them in 4 different wells. And so for our 10% pellet and our 0.5% pellet, we want 20,000 cells. So again, we calculated the 64,000 cells per mil. And so what portion of that do we want to actually put into our tube? So we've got our 2 tubes that are going to go into the centrifuge. What portion of our cell solution do we want? So we can do some real easy math. 20,000 cells divided by 64,000 cells per mil is 0.31 milliliters. So you might be using a micropipette to measure that. And that would be, so about 310 microliters in each one of these based on these calculations, right? So you're going to put it in the centrifuge, you're going to end up with a pellet. You're going to end up with a cell pellet. Then you're going to take off this supernatant here. So the reason why this step is important is because, remember, we're going to do the 10% condition and the 0.5% condition. To deactivate the trypsin, you're using this 10% media, but if you leave the 10% media on the cell pellet, then you're actually going to end up with a lot higher than 0.5% media, right? So we've got a cell pellet with the right number of cells in it, and then you can just resuspend. So we're actually going to end up with a higher volume of cell media with 2 mils and mix those cells back into solution. And then those 2 mils, you're going to put half a mil into each one of these wells, right? So you've got one tube for your 10% and you've got another tube for your 0.5%. So just some tips overall. You're going to observe the cells in your flask before you tryptanize them. So taking a picture in the microscope is going to be needed for your post-lab. Don't forget to do that. Don't forget to save your picture. So observe your cells, tryptanize your cells. So that means lift them out of the flask in solution. Take your sample for cell counting, and then decide what portion you're going to need to actually go into this well plate. Pellet the cells, and then resuspend in either 10% serum or 0.5% serum for each of the 2 tubes. And those are going to be plated in your 48 well plate, and we'll use them for analysis later. So hope you have fun.